So first of all, I would like to thank Professor Arijit for this opportunity. So in today's talk, I'll be talking about our research work on Demixer, which is a tool for detecting mixed infection microbial strains. So this is my talk outline. So where I'll be talking introducing the biological problem called as mixed infection and the approach that we have used to detect it. So microbes are everywhere. So we know that microbes are in the environment, in the food that we eat, and even in our body. So there are several studies that highlight the role of microbes in health as well as diseases. So like some microbes are pathogenic, where some microbes are beneficial. So we need to understand like what makes a microbe more pathogenic than the other. So the latest sequencing technologies provide a way to look into the genetic composition of microbes at a very high resolution. So in the recent times, we have different types of sequencing data, such as whole genome sequencing data that reveal the genetic composition of a particular microbe, whereas then metagenomic sequencing data, which has the genetic data of several microbes present in a particular environment, and transcriptomic data. So in our work, we are mainly interested in analyzing the whole genomic sequencing data of different microbes, specifically TB and TB. Okay. So this TB is a disease that has been in existence for several years, and it is caused by the bacteria, Mycobacterium tuberculosis. So treating TB in the recent times is much difficult than treating some years back. So one of the main reasons is that the treatment of TB requires the treatment period is so long for TB, and the patient has to take a combination of several antibiotics, and the things gets complicated if that particular case is a drug resistance case. So what is this drug resistance? So it is like the medicines that are intended to work for TB will not work as expected. So there are several reasons attributed to drug resistance, and one of the hypotheses is that presence of mixed infection could make the disease condition much severe. So here, let's see what is this mixed infection. So TB could be caused either by a single strain of TB bacterium or by more than one strain of TB, which is we call it as mixed infection. So consider this case in which a person is infected with two strains of TB bacteria. The green strain, let's assume that the green strains are drug resistant, and whereas the red strains are drug sensitive. And during detection, we may miss out the presence of the green strains as they are present in very low number, and the person will be administered with some antibiotic to kill the red strains. And things get more worse when the green strains are resistant to that particular antibiotic. So what happens is the number, after the administration of antibiotics, the number of red strains reduces, whereas the number of green strains increases, resulting in treatment failure. So this is an example to illustrate how we could detect mixed infection from whole genomic sequencing data. So consider a sample X, and here the sample X has mutations at 3 positions 1, 2, 3. At position 1, instead of the reference allele T, we have all the alternate, all the reads have the alternate allele A, and in SNP position 2, instead of, only 2 reads have the reference allele G, and the remaining 4 reads have the alternate allele A. And at position 3 again, 2 of them has the alternate allele T, whereas 4 of them has the reference allele C. So it indicates that some of the SNPs are heterozygous SNPs, indicating the presence of mixed infection. Now, if you are having the reference database of the known strains, let's assume that strain 1 sequence is AGT, and whereas strain 2 is AAC, then the proportion of the 2 strains present in the sample will be 33% and 67%. So there are several approaches to detect mixed infection. The computational methods could be put under 2 categories, namely reference-based and reference-free approaches. The reference-based approaches use a reference database of known strains, and they could determine the identity as well as the proportion of the strains present in the sample. Whereas in the case of reference-free approaches, they are simple probabilistic approaches that could reveal only the proportion and could not reveal the identity of the strains present. The problem with the reference-based approaches is they could not be able to detect the new strains. So some of the challenges that are not being addressed by the existing approaches are, suppose like if there is some new strain is present in a sample, how to detect it. And also another thing is that most of the existing approaches analyze only one sample at a time. So like if you want to do some comparative analysis, could we be able to identify any hidden information present in the samples? So our goal is mainly to develop a probabilistic model that could aid in the direction of novel strains in a microbial sample. So how we are going to approach this problem? So if we look at this, so here like our input will be a set of whole genome sequencing reads, and assume that suppose each of the read is assigned with a tag that gives the strain that is strain that is associated with that particular read. So if that information is known, then we could determine the proportion of the strains present in a sample very easily. And also we have some prior information about the known strains. So using that information, would we be able to detect the novel strains? So here, like this indicates that we could use a Bayesian approach to determine the mixed infection by defining some generative process for the whole genome sequencing sample. So now let's see the generative process to generate a whole genome sequencing sample. So if you see this whole genome sequencing sample, it is nothing but a set of SNPs, and each SNP, it will take either a reference allele, alternate allele, or some cases both reference and alternate allele in the case of mixed infection, and each allele in turn will be supported by a set of reads. Now so here, given a whole genomic sequencing sample, we want to determine the proportion of the strains present. And this problem is very similar to the topic modeling problem in text mining, where given a collection of documents, we want to find a set of topics present in each document in an unsupervised manner. So here, if we see, like the document could be very easily related to a biological sample, word to a sequencing read or a SNP, and then topic to the bacterial species, and corpus to the set of biological samples. So there exist several established approaches to for finding the proportion of the topics, such as latent Dirichlet allocation, non-negative matrix factorization, and latent semantic analysis. So in our approach, we are using the latent Dirichlet allocation method for defining the whole genome sequencing sample. So this latent Dirichlet allocation approach, it takes an input matrix X of size m cross n, while m correspond to the number of samples and n correspond to the number of features, and the output of this model will be two matrices. So this Y matrix, which is nothing but it will give the proportion of the topics present in each document, and the Z matrix will give the proportion of the words corresponding to each topic. So now if we take our strain analysis problem, it is nothing but it will be nothing but the proportion of the strains present in each sample and the distribution of the SNPs corresponding to each strain. So now I will give the overview of our methodology. So here we are having only the whole genome sequencing samples. We want to generate the sample SNP matrix, which is nothing but the count matrix, which will be given us to the LDA model. So we take the whole genome sequencing samples, then perform alignment and variant calling using the reference genome, and then construct the sample SNP matrix. And then another thing, though the strain analysis problem and the topic modeling problem are kind of very similar, there are some differences in the structure of the whole genome sequencing sample and the document. So we have made some modifications to the existing LDA model. So this theta and phi correspond to those two matrices that we seen earlier. So theta and phi are the parameters of the model that we want to infer, and Z is the tag that is associated with each read. And we also use the prior information of known strains, which will be given as input to the LDA model. And once the parameters theta and phi are inferred, they are interpreted to understand the significance of the mutations present in each strain. And then one of the contribution of our method is like the parallelization of collapse Gibbs sampling algorithm, which is used to infer the parameters theta and phi. So here, some of the data sets that we processed were very huge, and the Navy collapse Gibbs sampling algorithm was taking a large amount of time. But when after using this parallelization approach, we were able to reduce the time complexity from order of MNR to MN. So here M is the number of samples, N is the number of features, and R is the number of reads. And these are the experiments that we have carried out on different set of data sets. So this Biomix1, Biomix2, Biomix3, LDMX are the different synthetic data sets that we generated to check the performance of our model. And this in vitro data set is a data set that consists of some 48 samples like obtained by mixing real world TB samples. And so our method is also generalizable, that we were able to test it on a COVID data set. And these are two real world data sets that we tested our model on. So these are some of the results that we obtained using our model. So the first one is the result that we got on the simulated data set, where it could be seen that the method is equally well as the reference-based approach called as QuantTB. And then like in the in vitro data set, our model was able to determine the different lineages present in each sample. And then in the Malawi dataset, one thing that we also tried out was whether the model is able to infer the novel strains. So what we did is we hide the identity of some of the strains and tried to check whether the model could detect it. So there were some samples that was a non-tuberculosis, that was having non-tuberculosis strain called as Mycobacterium bovis. And it could be seen that, so these 5 samples are having like, these 5 samples are pure samples having the strain proportion 1, whereas this is a mixed sample having 2 different proportions. So, and the model was able to detect it. So the conclusion is that like our model is able to detect the strains with some 96% accuracy on different benchmark data sets. And also it is 7x faster than the Navy LDA approach. So it facilitates analyzing multiple samples together. But cons with, not con exactly, so though we are able to detect mixed infection, we could not find any effective correlation between mixed infection and drug resistance. So what we thought was like most of the samples that were having mixed infection will be drug resistant, but it was not we could not establish that in the data sets that we analyzed, because even the pure samples were found to be drug resistant. I'd like to thank my guide, Professor Manikandan Narayanan, for his valuable support and guidance, and Dr. Shivakumar from NART for his help in analyzing the COVID data, and our BIRDS and IBSC lab members for their valuable suggestions during the presentations of this. Thank you.
Wonderful, thank you. And that was pretty much well within the limit. So you have plenty of time for questions, and I'm sure there'll be many, given the kind of work that she has done. So open.
Very, very nice work and very nicely presented. So in the last slide, when you said that you didn't find correlation between mixed infection and drug resistance, so why would you expect that? Because in the mixed infection, let's assume, you're assuming that a drug-resistant strain and a drug-sensitive strain would be equally present, right?
Yeah.
What if the distribution is 99:1 or 99.9:0.1?
Yeah.
Right. So the drug-resistant one would be numerically very less to begin with,
Okay.
and over the time when you start the treatment, the sensitive ones gets cleared off, but the resistant one survives. And therefore, what you consider as a pure population as drug resistant could be outcome of a treatment, and as a consequence, removal of all the drug sensitive strains. So I don't see a dichotomy there.
Okay.
What you try to say, right?
Yeah.
So in the sensitive strains, you would expect mixed infection, but whether you were able to see the kind of the distribution. That frequency of the SNPs associated with drug resistance are very less?
No, sir.
Did you find something like that?
No, in some samples they were really high. Yeah, actually, like the distribution was high, but whereas in some samples it was very less.
And what were they classified phenotypically? Do you have data for phenotypic characterization?
Sir. There is yet another tool called as TB profiler that will determine the drug resistance using some mutations. Like it uses a curated data set.
That is genotype to phenotype, but for all the sequence analysis from whichever strain people have done, they would have also characterized functionally that whether they are drug resistant or not, right? Experimentally.
Okay,
so that tells you whether the SNPs that you are finding is actually giving rise to the drug resistance or not.
Yeah.
So if your distribution, the frequency of those strains are not more than 10%, you will actually classify them as sensitive.
Okay.
And not as a resistant one, while you are seeing the SNP in that.
Okay.
So that if your model is able to capture this distribution, that will be fantastic actually.
Yeah, sir. But like there are some databases that determines the drug resistance based on mutations. Are they like no, like like the experimental ones? Yeah.
Still, functionally, what you do on experiments and confirm that yes, these are drug resistant, till the time it is not resistant.
Okay. Yeah, sure. We will look into that. Yeah.
Perfect. Any other question for Brintha? Yes, here you go.
I have a question, and this is not really a question. I wanted to understand your method better. So when you say you start with reads, do you start with with contigs, or do you start with FASTQ files or FASTA files? Where in this process?
Yeah, we start with the FASTQ files, then generate the VCF files, then merge the VCF files of all the samples to generate the count matrix. So the count represents the number of reads like at a particular SNP position.
I see. So then it's independent of any method that one uses for sequencing. You can apply your algorithm to that.
Yeah.
All right. Great talk.
Wonderful. Any other question, just in case? Great. If there is no question, let's give a big round of applause to Onda.